Hello everyone, this is Dr. Vishal Trivedi from Department of Biosciences and Bioengineering, IIT Guwahati. Now, in a chromatography system, which is actually a integrated system where you have the multiple components, put it into a machine, and that can be used to enhance the efficiency as well as it can improve the purification. So, it has multiple components. You have a solvent reservoir. So, the component number 1 is a solvent reservoir. Ideally, you can have 1 or 2 reservoirs. In some cases, in some of the machines, you can have even 4 reservoirs. So, the purpose of having the 2 reservoir is that the first reservoir can be used to equilibrate the column, whereas the other reservoir can be used to generate a gradient. So, in that case, for example, you can fill the A with the buffer and the B you can use and fill it with the salt, and that is how, if you want, you can actually make a gradient between A and B with by the mixing these two, you can be able to make a gradient of this salt, and that can be used for the chromatography techniques. Now, these two reservoirs are attached to the pump. The pump, you can have 1 or 2 pump, which means you can have the pumps dedicated to A, you can have the pump which is dedicated to B. Different types of pumps people are using in chromatography system. For example, you can use the peristaltic pumps, you can use the syringe pumps, or you can use the infusion pumps, and in some cases you can use the high power and high pressure pumps. Mostly these pumps are based on the pressure level required to perform the chromatography, and accordingly you can use the chromatography at low pressure as well as the high pressure. So, based on the type of pump which you use in the chromatography, the chromatography could be of low pressure chromatography, where you will use the peristaltic pumps, or you can have the middle pressure chromatography, where you will use the syringe pumps or the fusion pumps, or you can use the high pressure chromatography or high pressure pumps, where you will use the pumps which are made up of steels. Now, after the pump you have the mixer chamber. The purpose of the mixer is to mix the buffer received from both the pumps to form a linear or a step gradient. So, the mixer is nothing but a kind of a magnetic stirrer. So, in the mixer what you have is a chamber which actually has a bead, and as soon as it receives the liquid from the two chambers or two reservoirs, it actually mixes them in such a way that it actually makes the linear or the step gradient, and then this liquid can be delivered into the column, and it can be used for the different types of chromatography techniques. As per your requirement, you can have the linear gradient, either the upward linear gradient or the downward linear gradient. Now, next to the mixture you have the sample injection port. Sample injection port allows you to inject your sample. So, in most of these chromatography system, you cannot be able to inject the material directly into the column. You can use this sample injection loop, and the sample injection loop is nothing but actually a valve where you can use a syringe to inject the sample, and that sample actually goes and fill into the loop, and then when you give the command to the system, it actually loads this material into the column. Now, next to that sample injection loop you have a column. A column could be made up of glass or steel. So, depending on whether you are using the low pressure column or middle pressure chromatography system or high pressure chromatography system, you have the option of using the column which is made up of glass or to the steel. Now, next to the column, now the chromatography is over, the sample is going to come out, and then it will be analyzed by a detector. So, the next component is detector. In the detector, it is actually a online monitoring system. So, the elution coming out from the column goes to the online monitoring system to test the presence of analyze based on the different properties. So, the detector could be UV visible detector. So, you can put UV visible spectrophotometers. So, you can have the UV visible detectors, you can have the fluorescence detector, So that actually will measure the fluorescence of the molecule which is coming out from the column, or you can use the RI detector. So that will actually is going to measure the refractive index, or you can use the molecular weight, or you can use the MALDI as a system. So in that case, this machine is going to be called as the LCMS, because it is actually going to do the chromatography, and at the end what you are going to see is the molecular weight of the molecules and so on. So you have the choices of different types of detector which you can put, and that detector is going to detect that particular physical property of that particular molecule which is coming out from the column, and then the detector will give that signal to the data recorder and analyzer. So in the data recorder and analyzer, you will see the profile of the eluent with respect to the measured properties in a detector, and it can be plotted in a recorder. This recording only we have called as the chromatogram. So the property is being plotted against the elution volume or the elution time, and that you can actually monitor in a online system. So you will see, as long as the protein is coming out, you can actually be able to collect the fractions. Now this detector is also connected to the fraction collector. So as soon as you will see that some compound or some product is coming out, you can actually ask the machine to collect that particular fraction, and that will be collected into the fraction collector. So in the fraction collector, you have the different types of glass tubes which are being kept, and these glass kept tubes are allowed you to collect that particular fraction or that particular protein into a separate tubes. Now this is all about the theoretical explanation of a chromatography system, but when you see a chromatography system in your laboratory, or when you see a chromatography system, the operation of these chromatography system is very different from the description just now I have given. So for this purpose, we have prepared a small demonstration of the chromatography system which is present in our laboratory, and the help of this demonstration we are going to show you how to operate the chromatography system, how to connect the column, how to connect the loops, how to inject the samples, how to, and as you know that most of these chromatography systems are being operated through a interface through computer. So you can also use different types of softwares to operate the machine as well. So with all these we have shown into a small demo clips.
In this video, we will show you how to operate a FPLC instrument and the basic principle laying mechanism of how separation of the proteins using FPLC. FPLC nothing but fast protein liquid chromatography. We can say it is a derived version of HPLC. The main difference between HPLC and FPLC is FPLC can only be used for the separation of the proteins and sometimes small molecules also if we have columns available. But in HPLC we can use columns for separation of small molecules. Suppose if you have enantiomers, that also can be separated using HPLC. The columns what we will use for HPLC and FPLC also differs. In case of FPLC, we will use plastic columns, but in case of HPLC columns, we will use steel columns. Because the main reason why we are using like this is, if you use stainless steel columns in FPLC, because of the salts and high concentration of the salts and different materials we are using, it may corrode the steel. So that's why there may be improper separation of the component which we want to separate. So that's why we will use only plastic columns in this one. The separation of the proteins through FPLC is based on the size and shape of the protein. So if you have, like in gel filtration chromatography or size exclusion chromatography, in all those mechanisms like size exclusion, gel filtration can be applicable in FPLC also. So it depends on what column you using. So if you want to purify histone proteins, you can use nickel NTA column, pre-packed column. Suppose if you want to only separate high to low molecular weight proteins, you can use any gel filtration column suitable for your protein. But the principle behind the separation is same. So these details of like size exclusion or gel filtration we are shown in previous video. In this video we will show how this instrument can be operated. We are in our lab we are using Octapure. This is from GE Health Sciences. So all the component whatever we show, it is similar in other instrument or other companies instruments also, but only the architecture of the instrument changes. So let's see what are the parts it contains. So this is the instrument. It is connected to a system for observation purpose. So in this system it contains this stationary phase. This is the column. This is the mobile phase, that means buffers. This is the area where we will keep all the buffers. So it starts with the pumps actually. So these two are pumps, this one pump and two pump, these two sets. Whatever buffers are coming from this buffer tray, it will enter here. Okay, these pumps, whatever the pressure they are getting, they can be regulated here. This is the pressure monitor. This. So once the pressure is monitored, it will go to mixer. This is the mixer where two different buffers. Suppose if you are using if you are purifying through nickel NTA column, so in that case you need imidazole in a separate buffer prepared, and one is the equilibration buffer. In that case, if you want to elute that protein, particular histone protein, you have to mix both the buffers A and B, for instance. So those buffers can be mixed here. Once the mixing is done, it will directly goes to the inlet loop. So once it entered into inlet loop, it will go to this chamber where it can be connected to the column. So the top portion of the column it will connect here. We will show you how to connect the column in coming video. So after that, whatever it comes, it will enter here. And it will come directly into the UV chamber where the eluted component will be detected. So starting the instrument, there is a power button right side of the instrument. You have to just turn on that instrument. Then you can see a white light is blinking here. That means the system just started. So after that we will go to this software part. For analysis of any elution, we can use this. It comes with the instrument. It is the Unicorn software we use for analysis purpose. So just double click on that one. And it will take you to the software. So it will give you three pop-up windows. One is method editor, another one is system control, and evaluation classic. So this is system editor where you can see chromatograms, and the other one is evaluation classic where you can analyze your chromatogram. Just go for system control. The first thing we have to do is connect the instrument. So here you can see the connected instrument Octapure 25. So just say OK. Now it will connect the system. So this will give different we can change different commands using this software. Just go to manual, execute manual. So this is the manual instruction software or dialog box where you can change things. So here different parameters you can change through this pop-up window, like pumps, flow path, and various parameters such as monitors. Just to go to pumps. So here you can change the system flow. So we can keep up to 20 ml if there is no column connected. So normal condition we can keep 5 ml also. So you just say insert this thing in order to execute it by system. Now here it is monitors, very important thing. We have 3 wavelengths here. We can monitor at 3 different wavelengths. So your choice you can give. So we are giving 280, 215 and 254. Just insert and say execute. So it started. You can see the green path is highlighted and also chromatograms appearing in the chromatogram area. So it will give 3 different chromatograms. So one corresponding to blue, that is 280 nanometer for tryptophan tyrosine fluorescence. Second one is 254 nanometer for RNA or DNA related, and third one is 215 for peptide. So here we can see the path of the flow, how it is connected, starting from buffer A. So here buffer A, and it will go through the pump and mixing. Through mixer it will go to all the way to waste. So here different parameters we can change during running. We can change B also. If you want to change B, you just say start pump B. So see, we can see highlighted area. If you stop the program, it will save automatically. See, you can see some dialog box appears preparing for new run. This is software introduction. These are the buffers we are going to use for this demo. The buffers need to be filtered through 0.2 micron filter and also degassed. For degassing purpose, we will use bath sonicator. So it will remove any air or air bubbles present in the buffers. It will remove those things. So we are using most common buffer, that is phosphate buffer with having pH 7.4, and this is the liquid water, and this is 20% ethanol. All the buffers were filtered through 0.2 micron filter paper and also degassed. So we have washed already, the system already being washed. So now what we will do, we will connect the column. So here precautions need to be taken while connecting this thing. So if you have any air bubbles through these loops or the piping system, it will directly enter into column, which will destroy it. So to prevent that, we have to make sure all the loops and pumps got washed thoroughly, and then we will connect in running condition. Before connecting the column, we have to remember few points. This column, whatever the bits are there, this is in 20% ethanol. So if you directly connect it, already ethanol is there. Whatever the flow rate we are giving, it will give more back pressure. So the bit between the the distance between the column filter and the beads, settled beads, may increase. So that will reduce efficiency of the column. So what we will do, we have to change these pumps into water. So we changed into water. Now we can connect it to the column. Here also some of the precautions need to be taken. If you are using chilled buffers, okay, suppose you need cold buffer, so that means you have to bring those all the components of the system to the temperature which you want to use for your purification. Otherwise, if you having a chilled buffer which directly enter into column, that may clog or precipitate some of the salts present in the buffer inside the column. So that will also reduce the efficiency. So this is also need to be taken care while running the FPLC. Before connecting the column, we need to adjust few parameters. So here is the software system flow. I am keeping 0.5 ml per minute since we are going to connect the column. So if the flow rate is more, then the pressure alarm may come. So after that we have to set the monitors. So this is also I am going to set. System flow 0.5 insert, and now we have to set alarms. At what pressure you need to get alarm. So I am keeping this 0.8 is fine. So once it is done, you can insert, then execute. So next here, this is the column connecting portion. So where here this is the upward portion where we have to connect with the column. Now we are not using column, so just we have to go to the column. We just click this one, column down flow. So from top to bottom, now you can see highlighted one. So you cannot directly connect to the column. First you have to fill the buffer or water in this loop so that there is no air bubbles. Just open the top of upside of the column. Okay, with this buffer itself you just directly connect. After connecting, you have to take out the lower portion of the column, otherwise it may burst also, but it is not the case, because if there is any high pressure, you will get alarm. So here this is the bottom portion of the alarm. You can see the buffer here if the buffer is passing through the column. So as we can see, there is a fill up of water in this thing. So once you see complete fill up of this loop or this knob, you can directly insert the lower portion. Once you see the buffer filling in, you just have to connect with the downward portion. So now the column is connected to the system and you need not to touch anything. Everything will be operated on the software. So here see, once the column is connected, you can see there is a change in the different UVs and conduction of the buffer. this is the red one is the, sorry, this gray one is the for conduction, and that green one is the concentration of B, and these are three different UVs. Now we are washing with the water. So after once completely we removed 20% ethanol, then we will equilibrate in the buffer. So the main purpose of the equilibration is, suppose if you are prepared your protein solution in a, suppose say phosphate buffer, so you have water, you are not equilibrated with the phosphate buffer, then you cannot expect good resolution or good separation of the proteins, and also the proteins may not be stable in the other condition like in water, so they may degrade or they may not be useful if you are interested in the enzymatic reactions. So that's why we always do equilibrate the column with the same buffer which our protein of interest is dissolved. So this will also helps good resolution and keep the intact of the structure of the protein. So we are indirectly we are providing similar conditions for the protein, so it will behave in a native condition. So we completely washed out the ethanol whatever present inside the column. Now we will equilibrate in the phosphate buffer. So as we can see here, the conductance is completely comes to 0, and we can see the completely flat line, flat signal corresponding to UV280. So that means there is no ethanol inside the column. In addition to that, we already washed 30 ml of water. So the total column volume inside the column is around 25 ml. So we also washed with 5 ml extra. So we can be sure that it is completely removed. Now what we have to do is we have to just pass the things whatever we set already without disturbing anything. So here we can see a pass symbol. You just pass, then come here and change the things to buffer. So once that is done, we will reset these things to continue mode. So we can see it is again activated. So we have equilibrated the column using equilibration buffer. So as we can see here, there is a stable line corresponding to UV280, and there is no other illusion coming out. So with this we can confirm that we equilibrated the column properly. So it's time to inject the protein solution and analyze it. So for injection purpose, this is the port where we are going to inject the solution, protein solution, and this is the loop. Whatever we inject through this injection valve, it will be stored inside this loop. The size of the loop depends on how much protein you want to inject and the column capacity. So we have 1300 HR column also. It is it can be used up to you can inject up to 1 ml. So in this column we cannot inject that much. If you want to inject, we have the company people might also give this kind of loops. So this we will connect as shown in here, and we will use for the injecting the protein solution. So what we will do is we have to set few parameters here. So here flow path, so injection valve you have to show it inject here, so insert this one. Okay, once that is over, we just have to insert the protein solution. And execute the command. So as we can see here, the chromatogram here, the protein is eluted. So at 13 ml of retention volume. So if you want to identify the protein molecular weight or determine the unknown protein's molecular weight, you have to run this kind of analysis. Like you need to be known that what is the protein molecular weight, known protein's molecular weight, so which is available actually, commercially available. So you have to take that protein and just inject. Based on that, you have to construct a calibration curve between log molecular weight and the KAV, that is partition coefficient, which is calculated based on the elution volume subtracted with void volume divided by total volume subtracted with the void volume. That will give partition coefficient. So whatever you will get a graph straight line, and based on that straight line you can get unknown proteins molecular weight. So that can be discussed in size exclusion chromatography video. Now we will show you how to analyze the results. So this is the software used for evaluation purpose, evaluation classic. So you have to go to file, open chromatograms. So you have to locate where your file is kept. Just open that one and say OK. So here you are seeing so many things. This one corresponds to pressure and conductance. So you can customize the things, like what you want to see in the chromatogram is only UV to IIT. So you just keep those things and remove all those things. Okay, and here also it is showing 380. You don't need 380, you want to take up to 65. So you just go and change the y-axis. So you can see this is the chromatogram. Peak is very sharp. So you can also integrate the peaks. So this is UV 280 integration. So just say so it will give the exact retention volume of the each and every peak, how many peaks are present. So here we can see 12.86. this is the major peak what we have. So with this you can analyze the results. So if you want to calibrate, you have to calculate retention volume for all the proteins whichever you are using for calibration, and construct the calibration curve between partition coefficient and log molecular weight. So with this you can identify unknown proteins molecular weight. So in this particular FELC demo, we showed you how the instrument works, what are the different parts, and what are the precautions we need to take while running the instrument, and how the software works, and how to analyze these things. So another point we forgot actually. This is the fraction collector. So while your protein is emitting, suppose your protein is emitting at 12, so started at 12, you want to collect fractions from that time onwards till the end of the emission. So what you can do, you can use fraction collector also from main window. This is the fraction collector, it will automatically moves. So here it contains the sensor, but you need not to touch anything. Okay, so in main system control, here fraction collector, there is an option for fraction collector. How many fractionation you need to be done, when you need to stop fractionation, and how much feed tube, fractionation outlet valve, all these things you can set there. In addition to that, you can also set system gradient flow. Suppose you want to elute a protein with the gradient, you don't know at what particular concentration of imidazole, if you are using nickel NTA column, or at what particular concentration the protein higher or lower molecular weight elutes. So with this you can just adjust the concentration and length. You have to give in suppose 60 minutes. So what system will do, it will over 60 minutes of time it will increase 0 to 50%. So you can do this one reverse gradient also. So first you will give 15 and time you can keep just suppose 1 minute. So from time when you start the system, it will starts with the 50% of the B and reduces to 0. So all these things will make you familiarize how the system works with the fast protein liquid chromatography. So hope these things will help you to achieve your goals in your research. Thanks for watching. Now we understand how a fast protein liquid chromatography works. So after using the instrument, we have to from buffer to water we have to change the valves, because if you are directly keeping in the 20% ethanol, it may the ethanol which are present in the buffer and the proteins or salts present in the column, they may get precipitated and clog the column. So it is a better practice, first you change this buffer system to water, then wash thoroughly, whatever the salts present inside the column it got eluted, then again you change to the 20% ethanol for preserving purpose. That need to be remembered for better performance of the system.
Now, the chromatography can be performed in two different ways, the partition chromatography or to the adsorption chromatography. In the partition chromatography, the analyte distribute themselves into the two phases, liquid stationary phase and the mobile phases. The major advantage of this chromatography is that it is simple, low cost and has a broad specificity. It is further divided into liquid-liquid chromatography or bonded phase liquid chromatography. The examples of this partition chromatography is cellulose, starch or silica matrix, whereas in the case of adsorption chromatography, the matrix molecule has ability to hold the analyte on their surface through a mutual interaction due to the different types of forces such as hydrogen bonding, electrostatic interactions, van der Waals, etc. And the examples are ion exchange chromatography, hydrophobic interaction chromatography, affinity chromatography and so on. So, the difference between the partition chromatography versus the adsorption chromatography is that in the partition chromatography the molecule is going to distribute into the two phases, but the molecule is not going to adsorb, whereas in the case of adsorption chromatography the molecule is going to be partitioned between the two phases. One of the phase will be the matrix phase, where the molecule is going to adsorb through the non-bonded interaction, for example, the hydrogen bonding, electrostatic interaction or van der Waals forces. So, with this we would like to conclude our lecture here, and in our subsequent lecture we are going to discuss about the different types of chromatography techniques and their usage in terms of the purifying the proteins or protein molecules. Thank you.